Okay. Hi, guys. Yes. What? Oh, yes, I can do that. That's what I forgot. Yeah, highly unprofessional, but it's okay. We'll fix it. Like this, like this. PDF. And where's desktop? No desktop. Okay, put it in downloads. File. Okay, I'll let you download it, and I'll talk at the same time. Right, so what did I want to say? So first of all, sorry for being a bit late. I had issues with Concordia transport, well, actually City of Montreal constructions. I didn't post your grades for assignment number 3. The reason for that is the assignment 4 was kind of not that well done for most people. There's only one person that got full points. And so if there's one person that got it, I can't really, say that maybe the class doesn't know what the hell is happening, and right? So I have to keep it that way. For assignment number 3, I have to reevaluate so that I'm a little bit more lenient, because, I mean, if you don't have the answer, I can't help you, but if you have an answer that's somewhat close, I can help out. Okay, so I'm going to try to make assignment 3 a little bit easier in terms of grading to compensate for that, okay? You're probably going to say I'm evil, but assignment 4, I should have done it at the beginning of the semester because it's a good lesson. And I know that, like what I'm about to say, I'm not saying that you guys are using ChatGPT or not, but some people are, some people are not. Some people are just using Google. Some people are not verifying the information. Some people still use the databases to find the genes and still got the wrong answer, and some people did get the right answer, okay? My point here is the dystrophin gene, there's so much published about it that it appears everywhere on Google, but unfortunately, I was expecting a little bit more verification on your side for this answer, right? The reason for that is, at this level, you learned how to use the databases, right? RefSec, NCBI, maybe UniProt even, Ensemble, whichever you learn, okay? So you could just go on those databases, do a little bit of filtering, and you'll find the biggest genes. The smallest one was kind of hard. So I gave points whatever you got closer to the answer or did not. So let's talk about first the use of ChatGPT and then go to the use of Google and then go to the database, okay? So ChatGPT, the way it works is that it learned from publicly available data. So it kind of guesses the next thing that it has to say. Obviously, if its data set contains thousands of papers that say that the dystrophin gene is the longest gene, then the predicted gene that's going to come out in that sentence is the dystrophin gene. But in reality, it's not. And the reason for that is data keeps evolving. ChatGPT does not have the data that exists right now, right? So we have a bigger gene, CNTNAP2. The other mistake that ChatGPT does is that it tells you that the gene is 2.4 million base pairs, the dystrophin gene, but in reality it's 2.2. So that's another mistake. Additionally,
but it says 2.4 in the studies as well.
Yeah.
Some of the older studies.
Yeah, exactly. And I'll come to the older studies because some people used papers from 1995.
Yeah, that's the one I got.
There you go. Some others used papers from 1990 something. So it's not like one person. There's a lot of people that did this. How can you talk about the longest gene when you don't even have the genome sequenced? Right? I mean, it makes sense now that I tell you, but maybe you didn't think about it, right? And then I tell you that the genome was completed in 2022, and there's still gaps in it. So technically, you would expect that maybe that gene that you found a long time ago is maybe not the longest gene, right? So when you're doing assignments like this, try to think about the course as well, because as we sequence more, we'll find other genes. It's just that we don't have them right now. Maybe CNTNAP2 is the longest gene and we'll never change it, but it's clearly not the dystrophin gene, okay? So that's the first part. The second part, to find the smallest gene, is very hard because you can Google for like 3 hours and still find nothing, okay? Some people talked about the testis determining factor. Some people brought up some random other genes, etc. There's a way to find the smallest gene, or at least some of the potential smaller genes, by doing searches on the databases. Okay, so that was the little independent research that you had to do for this. The titin gene was pretty easy. Is it because you know how to use UniProt, and you just went, or you Googled the name and you went on Medline and that's it?
There was a study that was like classifying the genes, and they had this table that you could download in Excel format. So
Yep.
I did that, and then I sorted them from largest to biggest using the Excel algorithm, and it just.
What year was that study?
It was 2021.
2021. Okay.
Gene size matters, analysis of gene length in the human genome by Younes.
Yeah, so it's not bad. It's very close, but not up there yet. I mean, it's very hard to give points when you don't really have it. So I try to accept, like for the small gene, I pretty much accepted almost anything that was in like the hundreds bases. If you went to the thousands, then I can't do anything for that. What else? For your explanations. So when you were telling me how confident you were talking about the fact that it was a consensus in the papers, right? Every paper had it, so it must be that, right? But then you can go deeper in the thinking and think that maybe those papers are outdated, okay? So this assignment was a little bit hard because it seems easy. Just do a little search, find a gene, that's it, and that must be it. But you needed to verify the information, and I think that's one thing that people didn't do, because if you technically consider uncharacterized proteins or uncharacterized genes, then you can find even smaller than that. There's 2 amino acid genes, like proteins, that are considered proteins, but they're not tested, so we don't know if they are actually true. If you look at the curated data, then you'll find that SMIM46 is one of the smallest genes that codes for a protein. So it was a little bit hard, I understand, but I think this is kind of a small little wake-up call to get in the habit of verifying what you put there, okay? So I looked a little bit at the dates of your publications that you were adding to the assignment. Most of them are in the 1990-something. That's not normal, because the genome is supposed to be sequenced in 2001, and then, one second, Gabrielle, and then from there, we only made updates to it, so we have new information. Okay? Yes, Gabrielle? Yeah, no, it's okay. It's good, it's good.
UniProt, right? It says, right, like the DMD gene is the largest known gene in humans, and it's 2.4 million base pairs. Like it says it.
Yeah. Look at the CNTNAP gene on the on NCBI's gene database.
Yeah, what's it called again?
CNTNAP2, contracting like protein or something.
I looked it up, and it is, like, I believe you, right? But
Yeah.
just on UniProt, it does say that it is.
I understand, but verify your information.
Yeah.
Verify your information. Yeah, it makes sense. Some databases, it's all, you know, if UniProt does not update their stuff. By the way, UniProt is not your source for nucleotides. Okay, it's NCBI. That's where you find it, or the Ensemble database. If you're looking for proteins, then you should go see UniProt. So they specialize in different things, but at the same time, try to verify it, okay? Being a little bit like harsh on this, I could technically accept the DMD, but I'll see with all your Honestly, what I was thinking is I'll see with all your grades, and if like I see that it's not going very well, I'll regrade this one. But for now, let's leave it like that. Okay. Yes, Matthew.
I was just going to say, you think it would be fair to say at this point that probably it's unlikely that we'll find a bigger gene, given that I don't know if there's any gaps left that are like
The CNTNAP. That's a complicated question to answer because there's 3 billion bases, and we have an idea of what ORFs are. Start codon, stop codon, things like this. But at the same time, there's so much overlapping stuff that I think it's probably going to stay the biggest gene because our computational power is so good right now that we can actually do all the possibilities. And if you look at the database, there's a lot of uncharacterized things, predicted but not actually sure about it. So maybe those will come out as all false, or maybe one of them will pop up as. But usually when something is big, it's easier to see. So if it's easier to see, then you'll have an easier time knowing that it's a gene. But if it's super small, that's where the gray zone gets into play. So for the small gene, I accepted almost anything. Like that's somewhat small. Obviously, if you told me. And the other thing I realized in this assignment, you had like the wrong sizes for the genes. The RPS, RPL genes. Now those ones, whoever answered those, that's funny because I work on those specific proteins right now, and they're not 200 bases. They're like 5,000. So I don't know how you got the numbers for 200 bases. RPL 14 is 5,800 something, right? So that's that. One person gave me a peptide instead of a gene. The MOC-C peptide. It's part of a gene, but that's just a peptide in the gene, right? So that's not, the full gene. Obviously, if I don't get it by then, hopefully I will get it in by then, but if not, I'll give you a little bit of an extension. Today, I'm going to talk about this. It's the last theoretical topic. Next lecture, we're going to start the interesting topics. Whenever I have time from now on at the end of the lecture, I will start talking about, I told you I will talk about the scholarships and all that so that you get an idea. So every time, so we'll try to finish always on time, but every time I'll give you a little bit of information. For the presentations, I'm going to send you a kind of some sort of like a website that you can just select, like just draw your basically date that you're going to be presenting. Okay, so I'll add all the teams. Yeah.
Are we still able to volunteer to go first?
Yeah.
Can we volunteer to go first?
Does your team agree though?
Yeah, we talked about it before.
Talked about it. Okay, team number 1. Do we have team number 2?
Let's be team number 2. What?
No. Okay, discuss.
Get it done fast so we can study for the final exam. It's just basic logic.
Okay, we're gonna vote on this after class, okay? All right, guys, functional RNAs. Sarah, I saw your email. Sarah, I will send the email. Okay, so we covered all this. Today I'm gonna cover basically what's left on the functional RNAs, and then that's pretty much it, okay? Do you have any questions for me up to now? No? Okay. Start by miRNAs. So I'll get back to miRNAs a little bit. I just wanted to show you evolutionarily that miRNAs seem to be appearing more and more as we become more complex. There weren't any miRNAs found in Saccharomyces cerevisiae, but as you get more complex as an organism, you require more ways of gene control and transcription control. And so you will have these miRNAs that start appearing as we go through the clades, okay? So quick slide. In the humans, there's about 1,000 miRNAs that belong to 70 different families, a little bit more than 70 different families. And they can target up to 60% of the human genes, okay? So why? How come just only 1,000 can cover up to 60,000? It's because some sequences are pretty much the same. And so they can target multiple transcripts, okay? And many genes have multiple miRNA targets. This is what I just said, but the opposite sense. So one miRNA can target multiple genes, and multiple miRNAs can target one gene, okay? So it's a two-way thing. miRNAs were the initial discovery that led to scientists trying to use it as a mechanism to shut down gene expression in cells. So there was this concept of antisense RNA. So if you have an RNA that transcribes for something, your mRNA for example, or a functional RNA molecule that you wanted to silence, then we were thinking that what if we can make an antisense RNA, get them to bind to each other, and that would prevent all the proteins from binding to it because it's a double-stranded RNA. So they tried that at the beginning. Sometimes it worked, sometimes it did not work. And this led to the development of, well, not really development, because it exists in the body, but it was kind of copied to do it exogenously, okay? So we can do it in the lab. sRNA is used by most labs that work on gene expression, and it's a way to silence a gene of interest. So if you're trying to study the effect of whatever gene, you silence it with sRNA, you have your control, and you compare whatever you want to compare, okay? Example, in the lab, I use it to kind of downregulate a protein that causes cancer, and then I see what I can do with that, okay? So this led to the field of RNAi interference. RNA interference, I mean, which is short for RNAi, and it's a way to inhibit gene expression through double-stranded RNA that is complementary to the mRNA. I know you've seen this many times, but it's important that I go over it with the specifics of this course. So the double-stranded RNA, you can add it to your cells at the lab. All you have to use is some sort of lipid bilayer that you put the siRNA in it, and then you get it to fuse with the cells. They release it in the cytoplasm, and then all this happens, okay? So the double-stranded RNA that you put in has the sequence that is the antisense for the transcript that you're trying to target, okay? And this will be bound with the RISC protein complex, which refers to the RNA-induced silencing helicase complex, whose goal is just to cut somewhere on the mRNA. So it breaks down mRNAs, okay? Before it gets to this state, your double-stranded RNA has to be cut into a smaller piece by a protein called dicer. So dicer cuts the double-stranded RNA and leaves only the part that is required to then silence your mRNA, and then RISC is the complex that will go bring that single short interfering RNA to the mRNA and then cut it at a specific place, okay? The mRNA is digested, and it's called a slicing mechanism, right? Which is a little bit different from digestion, but it just basically cuts in the middle, okay? So the structure of the siRNA, if you remember last lecture, I talked about the other types of functional RNAs, and they all had a hairpin loop or some sort of loop in their structure. For example, the tRNA had those that cloverleaf formation. For the siRNA, it forms a double-stranded RNA molecule. And so in that case, if you're looking for it in the genome, then you need to find the sequence that is fully compatible with itself and does not create those hairpin loops. It's usually 21 to 25 bases. Most RNA molecules that you will hear about, or oligos that are important for something, will be in this range. Any primer, any whatever, okay? The only exception is maybe the last two that I will talk about today. And the body also has, so the cells have this endogenous siRNA, but it's not as prevalent as the microRNAs, and it's used to suppress transposons and pseudogenes, okay? Your body prefers not to use siRNA because siRNA will degrade directly the message, and it's just an evolution from protecting the cells from viruses, right? And so if you are dealing with a virus, your goal is to kind of destroy everything. If you want just to control expression, then what you're going to do is use instead microRNAs, which are easier on the cell because they will just repress the ribosomes and get them off the transcript instead of breaking it completely. So Genes, pseudogenes, they can produce siRNAs. This is just an example here where you have a gene that does a duplication. So this is your homolog, paralog, whatever. And then the first gene will produce, so the good copy, let's say, will produce the mRNA. And then the other one will act as a retrotransposon because it has a retrotransposon in it that will affect its direction of transcription. And so it will code in the other sense. And because it codes in the other sense, then it will make an antisense transcript, which then makes your double-stranded RNA, and then dicer breaks it, and RISC complex binds to it to break down the mRNA. Okay. You can also form a hairpin through duplication of the same pseudogene. So this is actually what happens to make siRNAs in your body. Okay. So the retrotransposon is then duplicated, and then there's an inversion that happens, and then you're going to make this sequence here, which comes back on itself and forms the hairpin. Is this kind of clear? Okay. One thing that is weird, not that weird because I'll tell you why, but that is weird because the siRNA and mRNA, pathways, they use the same enzymes. So they have the dicer, they have the RISC complex, but for some reason they have a different mechanism of action. The only protein that is different between the two groups is DROSHA. DROSHA is the protein that is found in the nucleus that will take a primary mRNA and cut these overhangs here to turn it like this. Okay. And other than that, the rest is pretty much the same, and they both bind the RISC complex, and they both work on repressing translation, but they do it in different mechanisms. The reason for that, mRNA is not perfectly matched to the two strands of the mRNA are not perfectly matched because of this hairpin loop, right? And because of this hairpin loop, when they are bound to the RISC complex, they don't align perfectly with the RISC complex, and so the slicer activity is kind of not perfect. So instead, what happens is that the RISC complex will actually inhibit the ribosomes directly, okay? And without getting into the details of this, so mRNAs, they do degrade mRNA also, but not as well as siRNA, okay? And another function that they have that is different is the fact that they're able to remove poly A tails, and this is just how they are positioned within the RISC complex, okay? For the siRNA, they are a perfect match, the two strands, right? Because they are a perfect match, they align perfectly in the RISC complex, and because of that, they can align with the transcript of interest, and then the slicer function or mechanism of that RISC complex will happen. The other thing is that siRNA, for some reason, they're also able to participate into epigenetic silencing, mostly in plants, because the RISC complex is also able to bring the methylase to the DNA molecule. Okay, so they have other functions than just breaking mRNA. Clear? Yep. Okay. So let me talk about the protein protein complex that actually breaks the whole thing. So this is called this is the RISC complex, right? And the RISC complex has 4 domains. It has the PAS domain, which binds to the siRNA or the mRNA. You have the mid and end domains, which are just kind of polypeptides on their own that help orient the RNA. And then you have the PIWI domain, which performs the slicer activity. Okay? So there are many types. Argonaut is one of them, and these are just paralogs. PIWI and Argonaut are paralogs. Okay? This here, I think I just explained. So the siRNA, at the beginning, it evolved to block viruses, to kind of get rid of viruses. And if you want to get rid of a virus, you just want to destroy everything, right? So this is why it's more frequently used for degradation of the transcript, while mRNA is more likely to trigger the translational inhibition through removing the ribosome, etc., which I showed you in last lecture slides, okay? As I said, this is due to the positioning within the RISC complex, because one aligns it perfectly and the other does not align it perfectly, and because it does not align it perfectly, the slicer function is less efficient. Okay, PW proteins. So as I just said, the PW protein is the one that cuts. There's many of them, and there's kind of two groups, so there was probably a duplication at some point. And then the PW proteins are paralogs of the Argonaut proteins. Okay, Argonaut is the one that is used for microRNAs, and then there is also the PW proteins. The PW proteins, which is similar to the Argonauts, but the only difference is the type of non-coding RNA that they bind. So there's another type that was recently discovered, is the PW interacting RNA. Okay, mRNA, siRNA bind Argonaut, pRNA or piRNA or whatever bind the PW protein. Okay, these two, they do the cut, and they are paralogs. So the PW is very interesting because we're discovering more and more that it has a lot of functions related to cancer. And other than cancer, it was initially discovered in the germline cells to inactivate transposons, because you need to make sure that the transposons are not active in your germline to avoid passing on messed up genetic material. It's less important in other cells because, it's still found in other cells, but the fact that transposon inactivation is less important in other cells because there's a mechanism to get rid of, not so fit epithelial cells, let's say. Okay. There's just some historical information that was found in flies. It's starting to be linked to, so first it was in the germline, now we're finding it more in the somatic cells. But one thing is interesting about this is the fact that it's weird because we have the genome, we have the proteins, we have all that, but for some reason we missed this. And now that we found it, if you think about transposons and those crap proteins that I talked about in the past, there's a lot of them, right? And so if you're talking about transposons again in terms of the PW system, then there's probably a lot of PW proteins, but for some reason we missed them, right? So we're just starting to kind of figure out that there's a lot of these guys, okay? So the way it works, pRNAs are a little bit longer than mRNA and siRNA, and the way they work is that they don't really, they do have the cut function, to just prepare the small piece of PW interacting RNA, but their main function is to actually do epigenetic silencing. So they will go on the DNA, they will use the guide RNA, which is the pRNA here, which will bind to the region of interest, and then they will bring in these proteins that act that basically perform epigenetic silencing. Okay, so DNMT, for example, is a methyltransferase. If you guys remember from your other classes, the methyltransferases, they add a methyl group to histones, and that closes up the chromatin. Okay. Now there's over 50,000 pRNAs that are discovered recently. Okay, so they're found in intergenic regions, not necessarily like the mRNAs, which are found in the same transcript that they're trying to silence. Okay. These guys, they act on, if they're acting on the DNA, they will be in the intergenic regions to not affect the gene, and they will be used to regulate those genes. Okay. This part here is just showing you how the pRNAs get amplified, but not necessarily interesting for us. Okay. Okay. So a little summary between the three of them. So we have three different short RNA molecules that can silence genes. There's two of them that act on the transcript mostly. The first one is microRNA, which is the inherent, the naturally kind of occurring mechanism that is found on the cells, and this one is originates from the transcript it's trying to silence. Okay, it uses this Strasha protein that breaks these two segments here from the microRNA, and then exports it outside of the nucleus and leads to it being cut into a smaller piece by dicer. Then the RISC complex will bind only Obviously, there's a helicase here that will separate the two strands, and that will lead to a single-stranded microRNA bound to this complex here, which has the PAS, MID, N, and the PW protein. Okay, the PW protein is the one that does the cutting. siRNA or siRNA, short interfering RNA, is the same thing. The only difference is that it lacks this drasha thingy here and does not have this hairpin loop. Because it does not have this hairpin loop, then it will be much better positioned within the Argonaut protein, the complex, the RISC complex, I mean, and then it will have a better function at cutting, okay? The drasha is not present here because there's no overhangs that need to be cut, but the rest is pretty much the same, okay? piRNA, a little bit different. Again, same proteins. The only difference is the precursor that is being used, and that precursor, the precursor that is being used, and also the PW protein, which is able to recruit methyltransferases and acetyltransferases that are able to silence genes. Okay, this one acts mostly on DNA, and these mostly on RNA. Facile, easy, kind of. By the way, the details, all these details, don't go too crazy about them. The idea is just structure. Remember, just the structure. I'm talking about hairpin loops. I'm talking about these little things here, the overhangs, and then structurally, which one is more fit to do the cutting because it's just double-stranded, perfectly matched, while the other is not. And then, for example, on the exam, I might ask you, how would you find all the siRNA? Well, you would be looking for a sequence that when it bends on itself, it makes a perfect match. And to eliminate all the miRNAs, well, I will avoid all the ones that have a hairpin loop at the end of them. Things like this, okay? And if we're comparing this one with this one, then as I said, this one is a little bit longer than this one, right? So then you would be looking for similar structures in terms of RNA molecule, but this one is a little bit longer, okay? Functions. This is just a slide to show you the many functions that exist for piRNA. As I told you, methylation. For the PW system, I mean, it has methylation. It prevents it inhibits the retrotransposons, and it does so by just binding with different sets of proteins. For example, the line 1 retrotransposon is inhibited by the PW system. Okay. Did I forget something here? That's all for that. Look at that. I heard that you guys watch my videos in 0.75. Not going to say who said it, but if you want me to slow down, I can slow down, but then we're going to have an issue with the material that's covered. You know? There's so much stuff to cover, it's crazy. Okay, so long non-coding RNAs. So this is another group of functional RNAs, the LNC RNA, short for long non-coding RNAs. These ones are actually quite recent. We are just starting to discover them because of the next-gen analysis that we're able to do on the genome sequence. And it's a little bit different between these because it is basically super long and does not require proteins or things like this. We know some of their functions, but there are a lot of new ones that are coming up, okay? And when I say long, they're like 19 kb long, okay? So I'll give you an example by talking about sex determination, which is kind of interesting in this topic. If you remember from genetics, there are many ways to determine the sex of an organism. So you can do it either genotypically or environmentally, okay? I'll tell you just about the environment. For example, for reptiles, the temperature can affect the sex determination mechanism, okay? If it's too hot, too cold, that affects the reptile's sex determination. Some fish are actually able to change sex based on the social status. So who's the boss, who's not the boss, and who's the male or not the male, right? So they kind of switch up to make sure that there's an equal, not equal, but a distribution of sexes so that the kind of organism stays there and does not go extinct, okay? So that's for the environmental. We're not too much interested in that for now. What we're interested in is something that we're all aware of. So most sexes are heteromorphic. That means they have 2 different sex chromosomes. The XY for humans and fruit flies. Birds have this ZW system. Okay? So in mammals, if you ever take the mechanisms of development class, you'll learn a lot about this, the whole system, how it goes, and how you go from one cell to actually male or female. And so in mammals, the sex is determined by the presence of the Y chromosome. If the Y chromosome is there, it will produce this testis determining factor, SRY locus, and will lead to then male phenotype. If it's not there, and obviously inhibit the female hormones and things like this. If it's not there, then we can say the actual natural thing, or the natural state of the cells is leaning towards the female state. So if you know, so the basic state is female. If you have the Y chromosome, then you can become male. Okay, but if you remove the Y chromosome, the default is female. Okay. You've probably seen this. There are sometimes people that have triple of the chromosomes. Forgot the name of the disease. I keep teaching it, but keep forgetting them. So you can have many copies of the X, many copies of the Y, and then depending on the combination, you can end up phenotypically male, phenotypically female. But then obviously, for obvious reasons, you are probably sterile because you don't have the right dosage of hormones and things like this. Okay. There are also homomorphic sexes. For example, for the nematode C. elegans, they only have 1 chromosome for 1 sex chromosome, and it's the X chromosome. Okay. So how do we make sure that we're not expressing 2 times the amount of proteins from the X chromosome in females when we have only 1 set in the males? Okay, and obviously because we're talking about long non-coding RNA, and obviously that's one of the functions, right? But there are other functions that exist. So in organisms where you have heteromorphic organisms, there is always one of the X chromosomes that is either inactivated or something happens to it. Okay, so this way the goal is to basically have an even dosage of the X chromosome. Okay. If you don't do this, if the body does not do this, then you'll end up affecting the evolution of the species, and you'll end up interfering with actually the sex ratios in the population. What's up? And so I'll give you a few examples of how this happens. So dosage compensation, for example, in mammals, when you have in women, in female, you have the 2 X chromosomes, but one of them gets inactivated. Okay, and then the male just keeps his X chromosome. But if you look at Drosophila, the male's X chromosome actually is kind of transcribed and translated almost 2 times to compensate for the amount of proteins that are made. And then if you look at C. elegans, each X chromosome, if they're found both, if you have 2 X chromosomes, then what will happen is that they will be coded or translated less efficiently, and so you'll have less protein production per X chromosome. The way this works, for example, in this case here, if you have only 1 X chromosome, there's a protein in there that will kind of superactivate the translation machinery. And if you have 2 of them, it's just basically the amount of a certain protein affects how much transcription happens on that chromosome. Okay. In this case, if there's 2 X chromosomes, then the amount of a certain protein is higher than usual, and because it is higher than usual, it leads to the production of the RNA that inactivates the X chromosome. So there's kind of a threshold. Okay. So this RNA molecule, the long non-coding RNA molecule that leads to the inactivation of the X chromosome in mammals, is called a cyst, right? And if you take a look at this, this is just a staining of all the chromosomes in the nucleus. So you can see how it goes on top of the chromosome to prevent its transcription. Now, if you're trying to block the X chromosome from doing transcription and translation, you need something quite long. And this is the function of the long non-coding RNA molecule that is also, which is weird about it, it's spliced, it's polyadenylated, it's capped as if it was going to be translated, but then it's not. So this is something weird that we understand about it. And the reason why, so the X chromosome is obviously much bigger than 19 kb, right? But because it is condensated into a much smaller size, then just 19 kb is sufficient to inactivate the X chromosome. The other thing is this X chromosome will recruit these polycom repressors, which will come and bind to the two, so the chromosome and the RNA, and kind of make it hard for the translation machinery to do its job. Okay. This is a small image of this. So you have your cyst RNA. It has exons. It has kind of introns. It gets spliced. There's a poly A tail. There's a cap here, but it does not get translated. Or maybe it does, and we didn't find it. But you know, it's kind of weird. And what it does is that it goes on the DNA and just turns it into something like this. Yeah.
Is it always the same X chromosome in all the cells, or is it one at random?
We're coming to it. We're coming to it. Okay. Before I get to that, so this is what I was going to talk about. But before we get there, a very good question. Any questions up to now? Okay. So the X chromosome, when it gets inactivated in the female mammals, it will turn the it will basically make that chromosome kind of super condensed and turn it into kind of a heterochromatin state where it's silent, which is the silent and condensed form of chromatin. If you remember, euchromatin is open, and active heterochromatin is silent and condensed. And it creates these bar bodies, these black dots here that are seen under, I forgot what type of image is this, but this is only visible in female nuclei. We don't have this, well, you don't have this if you're a male, okay? So that's that. Now, which X chromosome gets silenced? This is weird, highly not understood, but depending on which X chromosome is being silenced, obviously you know that the two X chromosomes, one comes from the father, one from the mother, so the two X chromosomes are not exactly the same. And because you're expressing only one of the two, depending on which one gets activated, you'll end up having traits that are similar to mother or father. That's the first thing. And so there are, so as the cells are dividing, early on there's while you're still in the zygote stage, the X chromosome gets inactivated. Sometimes it's the paternal, sometimes it's the maternal. The choice is not clear how this happens, okay? But both of them are possible. And because both of them are possible, then you end up having expression only from, let's say, the X chromosome from the father or from the mother, okay? One very interesting example is the calico cat. These cats here, if you see these colors here, this is because of the X chromosome inactivation. So what happens is that there's kind of a shift between the X chromosomes that are being inactivated as the cells are dividing. And because this happens, then you end up with, for example, let's say if the father has the orange kind of color, then you know that this here is the mother's X chromosome that is being inactivated, and here is the father's X chromosome that is being inactivated. Make sense? So now when you see these cats here, you know where they come from.
One
Okay.
of the first things we were ever shown when I studied my biology degree in Concordia. First
Yeah.
class I took, they showed us.
We keep showing the same examples because we're running out.
Well, the cats are just cute.
Yeah.
It's striking.
Yeah. So did you guys see this before? Okay. So basically you don't need this class. Good. So tomorrow exam. Okay. This one is kind of complicated. So we have, sorry? This here? Yeah, no, there's no way you see this. Okay, there's only one slide on it. I don't want to get into it, but I talked last lecture about the signal recognition particle. Very quickly. So the signal recognition particle, if you remember, it was mentioned when I talked about the ALU element, the retrotransposon. And so the eukaryotic signal recognition particle is a complex of RNA molecule, the 7S RNA, which is a non-coding functional RNA. That is 300 nucleotides. I don't know whether it's the N here, but on the keyboard it's close to B, so I guess it's B. And there are 6 proteins. The 6 proteins are these. You don't need to know them, but basically what this does is that it binds to hydrophobic amino acids that are found at the N terminus of proteins that are going in the secretory pathway. Secretory pathway, so lysosome, exocytosis, things like this, okay? And the role of this RNA is to basically direct the whole SRP complex to this protein here, okay? Obviously, if the structure here is a bit weird. What I just want to mention is if you are looking for this structurally, then you'll have to enter all these different features from this SRP RNA to be able to identify it, let's say, computationally. Doing it using wet lab research is a bit more annoying and complex, so better do it, better do it computationally. One interesting thing, though, is that the ALU element that we talked about comes from this SRP RNA. We don't know really why, but part of it is part of the ALU element. Okay. Don't need to know anything more. This is the only thing I want to mention about this. And then the last thing I want to talk about is the telomeres. So this is a cool topic. You guys probably read this at the same time as people talking about immortality or talking about aging, right? So telomeres are quite interesting because it's something so simple but yet has so much effect on the cells. They're basically the ends of chromosomes and they protect the chromosomes. So the sequence of the RNA so obviously I'm talking about the RNA here because there's an RNA molecule that is part of the telomere system, telomerase system, and it has an RNA molecule that its only goal is to provide the kind of blueprint to make these telomeres, which are short tandem repeats of DNA. Each organism has a different sequence, but they're very similar and very well conserved, and obviously this underlines the important role that they have in protecting our cells from the breakdown of the chromosome. So you can see that they are TG rich. That's one of the features, and usually you have, for example, this one is very similar. Okay. Okay. So biology 261 review. When you have DNA replication, what happens is that you have 2 strands. So you have the leading strand, which is copied in a single run, and then you have the lagging strand, which is copied in multiple events. And the reason for that is just the direction at which the polymerase is going. So when you're on the leading strand, you have your you have your primer that is bound there, and then the polymerase recruited here and just goes on in this direction. But because you're going in the opposite direction for the lagging strand, you will basically go on for a little bit and then get off. And so what happens is that you will leave these overhangs, these kind of holes here that need to be filled. Okay. Now this is not a problem because the DNA repair machinery will fill these holes here, and that's okay. But there's one of them at the end that remains that is never repaired, and it's because this one, there's no primer sequence here to allow for the RNA polymerase to bind and start going this way. So the cell recognizes this as DNA damage and comes and starts eating up here. Next replication cycle, you create another overhang, but now you're playing in this region here, which is, let's say, gene-rich. You leave the overhang, DNA polymerase cannot bind to it, so there's a degradation that happens here, and then you go on until you eat up the whole chromosome. Usually the cell dies before that, okay? So what we're interested in this course is the telomerase. There's an enzyme in the cells that allows for protection of the ends of the chromosomes called telomerase. And this telomerase, what it is, it's just a reverse transcriptase. And it's a reverse transcriptase that is bound to a functional RNA molecule called the telomere RNA component, TERC, telomere RNA component. So when you leave the overhang, there is this RNA molecule here that will come and with the telomerase and bind to the overhang, which is of known sequence, and then will basically just extend it, right? So you come here, you bind to the overhang, the telomere, then you will start extending it. You move on, you elongate, you translocate, and then you keep adding more and more to the DNA molecule. Then when this goes off, you'll have a longer overhang, right? And then you can actually have primers that bind here and DNA polymerase fixing this piece here, okay? Here's the base by base example, right? So there's a primase that can bring a specific RNA primer that is quite short, and then the DNA polymerase will come bind to that. And obviously this primer here is complementary to part of the telomere, and then the DNA polymerase will come in and fill in the gap. Okay? Simple. So telomerase RNA, obviously you have just this little piece here, but for some reason when I show you the structure is this big. Because it has structural domains that are necessary for aligning it within the telomerase. And so obviously it's not just a little piece of RNA, but the piece of RNA that we're interested in is found in the pseudoknot here, okay? So the telomerase RNA is called TERC, telomerase RNA component, and it's just serving as a template molecule to increase the size of the telomeres. Okay, so this I just said. Something a little bit interesting, and then I'll talk a little bit about the scholarships, okay, because we have a little bit of time. So telomeres are very interesting. When they were discovered by Jack Szostak, whatever, his name is very hard to say. One of the professors here, as I told you at the beginning of the semester, was in his lab when he got the Nobel Prize. That's Malcolm Whiteway, the professor that designed this course. And so when they were kind of discovered, people were saying that basically we can be immortal if we increase the size of these telomeres. And we can do that. The only problem is that when you increase the size of those telomeres, you prevent degradation of the DNA, but you allow the cells to accumulate damage. Because cells, when they are growing, they have metabolites that are being used, and then they have waste that is created, and at some point the cell will reach a point where the repair mechanisms will be overwhelmed by the damage. And also the repair mechanisms in the cell will be damaged themselves, right? Just because of entropy, because of the way things happen. And so you do reach immortality, and actually this is a way that we use in the lab to make cells that we extract from organisms like mice. We can turn them to immortal by adding a telomerase and making it hyperactive, okay? And if you go work in a lab that has cell culture with human cells, then you'll see that you can just culture those cells, split them for your whole life, and they'll still be alive. At some point, they will start like detaching and dying, but it takes a lot to get them there, okay? Remember the Hayflick limit? The limit of times that a cell can divide before it starts being kind of damaged, and that's about 4 T cell divisions, okay? So in reality, what happens is that you'll end up having your telomeres, you'll have attrition of the telomeres until you reach a point where it's very short. This will signal the cell to stop dividing and will enter it into a stage of senescence. So senescence is the cell is still kind of living, undergoing metabolism, things like this, but it does not divide anymore, okay? One thing that is interesting about this stuff, which probably will be my lab's focus at some point, is using senescence to cure diseases, because each disease has changes in the proteins that are in the cells. And so when you have, for example, cancer, there are proteins that are upregulated or downregulated. Imagine you can use senescence to stop the growth of those cancer cells. Okay. Actually, I'm working on this right now. The last paper before I leave the postdoc, hopefully. So this is one of the work that is happening. Telomerase is found in all your cells. It's highly active in human tumors. So you can see why, obviously, because the tumor cells need to be immortal and they need to divide an unlimited amount of times. And so you need this mechanism here. It's detectable in normal cells, but not really necessary because these normal cells always get replenished through the stem cells, so you don't really need to keep them alive forever. What you need to keep alive forever is your germ cells and your stem cells, right? You need to keep them there for a while because if you run out of stem cells, well, that's it. Okay. So with that, that's the lecture for today. Questions?